To process information, cells need the right proteins for the right job. In case study number 6, we asked the question, which cells respond to steroids? Now, you need to understand that steroids, like the two shown here, are hydrophobic molecules, which means they can penetrate every cell in your body. So in this animation, we show that the steroid, because it's hydrophobic, can cross through the plasma membrane into the cytoplasm of any particular cell. In fact, that happens for every cell in your body. So when you have a steroid either released naturally or injected into your body, every cell in your body can respond potentially to this added compound. The question we want to understand is, how do we know, or how do cells know if they should respond by changing gene activity? Which cells respond? As you can see here in the animation, the one down at the bottom has changed its transcription level, changed its gene activity, as indicated by the blue color. Steroids by themselves cannot change any gene activity. Steroids must bind to a receptor, and the receptor is in the cytoplasm of the cell. And so, what kind of molecule is that steroid receptor? Investigators wanted to understand the kind of molecule so they might be able to understand how it works. In this experiment, the investigators pre-treated all the cytoplasmic molecules, everything that was in the cytoplasm. They pre-treated it and then asked the question, how much steroid could bind after the pre-treatment? And the negative control, there were no enzymes added to the cytoplasm, and you can see all the steroids bound that were possible to bind, and they set that artificially to say that's 100% possible binding. In the next experiment, they added pronase to the cytoplasm. Pronase digests all the proteins. Then after a while, they added the radioactive steroid and determined how much was bound, and you can see only 25% was bound. When they did the same experiment, but this time instead of pronase, they used RNase, there was no significant difference in the amount of steroid bound to its receptor. Likewise, when DNase was added, almost the same amount of steroid was bound to its receptor. So from this experiment, they determined that the steroid receptor is a protein, and they recognized that the receptor functions as a transcription factor, and it does interact with DNA, and maybe that's why this level for steroid binding in the presence of DNase was reduced. But that's uncertain. The main point being that the steroid receptor is a protein. Then they wanted to ask the question, where in the cell is the steroid receptor? Now you have to realize that y-axis on the left is measuring the amount of cytoplasmic progesterone, because progesterone steroid is radioactive. That's what they could measure. On the right, they measured how much progesterone was inside the nucleus. Both of these measurements were done simultaneously over a continuous time span of 30 minutes. At time 0, they added the steroid to the cells, and then they isolated a subpopulation of cells, blasted them open, separated the cytoplasm from the nucleus, and measured the location and the amount of the progesterone. You can see that over time, the amount of steroid receptor was high initially in the cytoplasm, but then it goes down over the 30-minute time period. Conversely, the amount of steroid located inside the nucleus was low, but it went up over time. So in fact, the steroid receptor appears to move from the cytoplasm to the nucleus, meaning that it didn't have a single location. It depended if there was steroid bound to the receptor or not. Once bound, it moved to the nucleus. So the steroid penetrates the cell and binds to its receptor. There's an allosteric modulation so that the receptor changes shape, and then after the shape change, it migrates into the nucleus. The receptor bound to the hormone acts as a transcription factor. The receptor alone is not a transcription factor, even though it's a protein, and the hormone itself, the steroid hormone, is not a transcription factor. Only when the two interact and locate in the nucleus is this a transcription factor. In case study number 6, we come to the conclusion that all the cells that express the protein steroid receptor were able to respond to the steroid. Even though the steroid penetrated every cell, only those with steroid receptor were able to change gene transcription.